My name is Jonathan Legrand. I'm an engineer in biological data analysis. So what I'm doing is to create tools and create code, helping biologists to go from these images they acquire to this labeled image, from which we extract cellular features, and from these features we do statistical analysis. Okay, so on this board we describe the pipeline we are trying to set up for this image analysis. So we're starting with potentially 5-dimensional images. So you have the XYZ spatial dimension, then you have channels and potentially time. So with these images, they go to a reader. This reader create a specific data structure. So for example, if you have a multiple observation of the same object from different angle, what you would like to do is to reconstruct and fuse together this multiple angle, and then that gives you one bigger image that you may want to filter. And from these images you can then perform segmentation, for example nuclei detection or cellular segmentation. For example, this is a polar transport pump. Being able to quantify how many pumps you have gives you an idea of the flow of protein or hormones. So from that you will be able to build up models and to try and make a realistic model of flower development or phyllotaxis, if you talk about flower arrangement around the stem. So compared to my original work, the WOMI project is for me represent a change in scale. So we're moving from the cellular scale or tissular scale, and where we try to understand how flower or leaves are arranged around the stem, to the microscopic scale where you see the plant in full and you're trying to observe also and quantify also how flowers or leaves are arranged around the stem.
So I'm a bit at the end of the pipeline. So as input we take a 3D point cloud, which is a sample of the plant represented in a virtual way. So we've got points which each of them has 3D coordinates, and only from this set of points we try to infer the geometry of the plant, and from this geometry, so basically to recover the shape of the plant, we try then to segment the plant into its organs, so the leaves, the stems, and in between stems and leaves, the petioles. So the idea is that only from geometry and maybe some color information or other information, but we try to use as less information as possible, we would be able to detect the organs of the plants and then do some computation, for example simple computation like computing the number of leaves, but also more advanced computation like, for example, trying to guess what is the area of the leaves or the angles between the different stems and so on, things that are useful for biologists and also for people in agronomy or agriculture.